this right here, this first section, this is the non-disjunction and karyotyping section. Okay, remember on this section, we're looking for either autosomal or sex-linked disorders. Now, there's only one autosomal order that you're going to have to know by name, and that's the triplicate in number 21 here. And if you've got 3 chromosomes in number 21, that is Down syndrome. And that's the only autosomal disorder that you have to know by name. All the rest of them, you just say there's an addition or deletion in chromosome number whatever it is. Okay, with these sex-linked disorders, there are 4 of them that you need to know. There is Turner syndrome, which is a deletion of either an X or Y chromosome, so that you only have one X chromosome. The next one is we don't have one represented on the table here, but it's called Klinefelter syndrome. And Klinefelter syndrome is the XXY. Oh no, wait a minute. We have it right here. Klinefelter syndrome. You can see here, and remember, you could get either this karyotype or this karyotype on the test. So be sure to be able to read both of them. But on Klinefelters, you get an X, X, and Y, and you'll need to know the symptoms of that. General symptoms, I can tell you right now, it's going to be a male because there's a Y chromosome. And they're usually a very effeminate male because they usually have a little bit higher level of estrogen in their system. Okay, the next one is the poly-X or triplicate X syndrome. And that would be the triple X. And that's going to be a female. And again, you're going to need to know the symptoms off that one. The last one is Jacobs syndrome. Now, for y'all's purposes on the test, the only thing you need to know is it's almost asymptomatic. That extra that I told you about, that they're actually doing studies to see if the extra Y chromosome causes a higher level of testosterone in the system, which causes higher levels of progression. That was extra information. You're not going to need to repeat that on the test. Okay, now for each of these, along with knowing what syndrome it is, you also need to be able to read the karyotype and know whether it's male or female. Let me make sure that's it. Okay, it also says here, know which chromosomes are autosomal. Which ones are they?
1 through 22. 1 through 22.
1 through 22, and which ones are the 6 chromosomes?
23.
No, 23. Be able to sex, disorder, identify the type. Okay. I'll do that one after this. Okay, Hardy-Weinberg experiment does this equation.
That color looks so prettier on this.
Okay? Now, the equation that they don't give you in the book, but I'm going to give you right now, is, and this will make it a lot easier to do calculations. Now, the way that this is set up, P square is your homozygous dominant genotype. So that would be like big P, big P. Okay? 2P square is equal to big P, little p, and then Q square is little p, little p. Okay? I'm going to erase that, but just keep those in your head for this. Okay, now, the easiest thing to find whenever you're doing population studies is the recessive genes, because you know it's just on the phenotype, it's little p, little p. So, what we did yesterday, or not yesterday, day before yesterday, was testing the PTC paper, and whether you could taste it or not If you couldn't taste it, you're the recessive phenotype. So we ended up with numbers like 16 to 5, or excuse me, if I remember correctly in this class, we got 11 to 5.
It was 10 to 7.
10 to 7? Pretty close. 7. Okay, now, what they're going to do in here is they're going to do it exactly like this. They're going to go ahead and give you P. And if they do that, all you have to do is plug it into this equation. So Q would be equal to 1 minus P. So if it's 0.8, it's just 1 minus 0.8.
Which would be 0.2.
Yeah, 0.2. And then all you do is plug and play. You take these numbers, plug it back into here, and then it's going to be like it doesn't have an answer to it. You plug it in to see what answer you get.
Yeah.
If it's close to 1, that tells you that since the equation is close to 1, there's no evolution happening in that population. If it's less than 1, then some sort of evolution is happening. So if it's close to 1, these equilibrium or no change rules here, the no natural selection, no gene flow, no mutations, no genetic drift, and random mating is all happening. Okay? If it's less than 1, that means that there is some sort of evolution change happening. Either there is natural selection, could be gene flow, there could be mutations, or there could be genetic drift. And there's also non-random mating. Okay? Now the farther away from 1, the more of these that probably apply. If it's a little bit away from 1, there's probably 1 of them happening, maybe 2. If it's a lot away from 1, like it's close to like 0.1, 0.2, then probably all of them are happening.
Okay, so the point, once you get to 0.2.
It's plug and play. You just basically take
0.2.
0.8
Right.
squared plus
Okay.
2 times 0.2 times 0.8 plus 0.2 squared.
Okay.
And I think if you do the calculations on this, it gets to be pretty, I think it's like 0.9.
Okay.
But,
Pretty simple. Okay.
but here's the thing. You don't have to do the calculations in your head. You will be provided a calculator during the test. Okay?
So they're gonna get, basically they're gonna give us the P and we just plug it in
Well,
with your.
they may give you the P or the
They'll
Q.
give us one or the other.
Yeah.
And all you'll do is change
Change
that
it.
Yeah,
first,
you
that
just need to
equation there.
remember this
It'll be
equation.
P equals 1 minus Q instead.
Okay, all right.
Yeah, just remember that equation because it's not in your book.
Okay.
Okay.
So is the
Charged, and alcohol has no charge to it. It's not water. It drew the DNA up out of this solution into the alcohol. And then at that point we took the hook, grabbed it out, and then looked at it under the microscope. As far as I know, we're probably not going to have the wet mount slide of the DNA on the test. You will just have this. Okay. Okay, next one is gel electrophoresis. I'm going to kind of just skip this because since your report was all over it, you should know all about what's going on right here. Okay? The only thing you may need to add to it is know what PCR is, which is what?
Protein chain reaction.
Yeah, polychain reaction. That's where we take the DNA and we duplicate it several million times so we have enough DNA to actually run the test with. The only other thing that I'm not sure if I told you, do you remember the liquid on top of the gel? Yeah, that was called TBE buffer. And for some reason that always pops up on the test. I don't know why. The second thing you'll need, last thing you'll need to know is how is the DNA strand separated. It's done 2 ways. It's 1 by size and then 2 by charge. Kind of remember that example I gave of the semi and the motorcycle going down the congested highway. Okay, the next several tables is just evolution. That's that lab number 19. I would make sure, if you haven't done it yet, take pictures, make drawings of everything on this table. Okay?
She lives in Colorado, so.
Now, I'm going to take you through these numbers first. Mm. 723. Now, this is the class numbers that I got
These are the
for one of the
current,
classes.
right?
Yeah, this is the number of current. So this would be purple smooth, purple rough, yellow smooth,
Yellow rough.
and yellow rough. Okay? Now, you start out. Would someone mind grabbing me the calculator off that back table? But what you're going to start out by doing is taking all of these numbers and adding them together to get a total kernel count. Okay? And if I remember correctly, not total kernel count. Yeah. Total kernel count would be 3,836. Okay? And this is actual. And this will be faster if I... Okay, you're going to take this number, this total number that you get, and you're looking for a 9 to 3 to 3 to 1 ratio. So you take 9 divided by 16, because that's how many squares you have on there, and you get 0.56. And then you take that and you multiply it by your total number, 383 times 3836. And this gives you 2158. This is what you should have gotten if you had a 9 to 3 to 3 to 1 ratio. And then you end up getting 719, 719, and 240. This is the numbers that you should have actually seen. And then what you do is you take the difference of this. Difference on this one would be 1. Difference on this one would be 4, 25, and 20. Then you go through and you square the difference. So this would be 1, 16, 625, and 400. Then you take the d squared, this number, and you divide it by the expected, which would be this number. So on this one, you get 0.005, 0.022, 0.869, and 1.667. Okay? Now for your chi-square number, you take all of these numbers and just add them together. And then that ends up as 2.559. Okay? It's just a lot of math.
Yeah.
The difference of the two, and then you square the difference, and then you divide it by your expected number, and then you add all those numbers together. This is your actual chi-square number.
What is the chi-square number? I guess
It's
that's what I don't
It's
understand.
a statistical probability of are your differences far enough apart to be significant. And then what you do is you take this number and you go off of, where's the chart? You'll be given this chart during the test, but you go off your chi-square chart, and since we're looking at 4 different phenotypes, we're looking at purple smooth, purple rough, yellow smooth, and yellow rough. That's your C value. And then you take C minus 1, that gives you the row, which is 3. And then all you do is you take this number here, the 2.559, and you look for it on the line here. And it fits in between 2.366 and 3.665. So that means it's here, and the differences are insignificant, so your hypothesis is supported. Or you got your 9 to 3 to 3 to 1 ratio in a small enough difference that it's significant.
Wow.
Yeah, no, that's probably the most complicated thing on here. Females are circle, squares are men on the pedigree chart. Okay. For everything right here, this is all the Mendelian genetics. Okay, that was counting the kernels of corn. Remember, some of y'all had trouble doing the dihybrid Punnett square. If you had trouble with that, just review it because you're probably going to have to look at that
Excuse me,
on
sorry.
the lab practical. Excuse me. Okay, you'll need to be able to know which phenotypes we were looking at. And the two that we looked at were color and kernel shape. Okay, the color could have been yellow or purple. The kernel shape could have been smooth or rough. Expected. Okay, the only other thing you're going to have to do on this one is review how you did your chi-square test. Now, for the most part on the practical, they don't make you have to do all the calculations that we did in class. They will give you a number and then you'll just have to know how to work it on the chart here. Remember, if we're looking at two different alleles, color and shape, that means there's 4 traits we're looking at. So those 4 traits would be equal to C over here on the left-hand side, and then you just take C minus 1 to find the row that we're looking at. So for the kernels here, it'd be row 3. The other thing you're going to need to know about this is F1, F2 generations, pretty much how the box looks here. Just understand that the colorations here is the F1 generation, and then we add kernel shape here, and that ends up being the F2 generation. All right. Moving on to, we're going to skip this for now. This is it. This is our DNA isolation experiment. We're going to move on to viruses and then we'll come back to this. Okay, for these, they were just the 2 handouts we gave. This is your HIV virus and this is your bacteriophage of the different stages. Now, this is the one thing that usually catches a lot of students on the practical. They'll give you, like say, this one. And then they're going to ask you, what stage is this? Now, we don't have numbers or names like we do for mitosis. All you have to do is describe what's happening in here. Like on this one, we would say that the DNA has been inserted and replicated inside the bacteria. And for the most part, when we get to that, if you're somewhat close on your description, I'll usually give it to you. Do y'all remember the word we use for what happens to the cell whenever it explodes like this? Starts with an L? No, lysis. That's the only word I require you to use on the description here is make sure you use lyse for the cell whenever it explodes. On the HIV virus here, you need to know the RNA, which is this little shoelace here in the center. You need to know the reverse transcriptase, which are the 2 purple balls here and here. So those are here and here.
Where are those?
Reverse transcriptase. The protein coat is this blue line right here. Okay? The protein coat. And then we have a membranous envelope that's on the outside of that. And then the last thing, which is the way the virus interacts with your T cell, are these glycoproteins on the very outside of it. Okay, DNA isolation. I'm going to just go over the experiment that we did. Remember that in this section, we also had a lot of book work that we were supposed to do on transcription and translation. So I would just highly suggest reviewing that. It's almost the same stuff that we went over during lecture. Okay, for the experiment here, we took the source of the DNA was your banana. We took that banana and we stuck it into the mortar and pestle, and we added sand and DI water to it. We crushed that up until we got that like pea soup consistency. We then poured it through the filter paper here, through the funnel into the test tube there. Now, what were we doing with this entire process that I just described?
Separating the DNA.
That's breaking the cell walls. And we were separating the DNA, but specifically what they're going to be asking for on the test is you're breaking the cell wall. Okay? Now, once we got it in here, what did we add to it? What was the first thing we added to it, I should say?
Soap.
Soap or dishwashing liquid, Dawn soap, however you want to say it. What was the purpose of the soap?
Break it down.
Yeah, breaks down the nuclear envelope. So that way it releases the DNA out into the solution. Now, what was the last thing we added to the top here? It was inside the green thing on the table up there, in the ice. Isopropyl alcohol. And it was cold 95% isopropyl alcohol. So, once we did that, because the DNA is negatively charged and alcohol has no charge to it, it's not water, it drew the DNA up out of this solution into the alcohol. And then at that point we took the hook, grabbed it out, and then looked at it under the microscope. As far as I know, we're probably not going to have the wet mount slide of the DNA on the test. You will just have this. Okay, next one is gel electrophoresis. I'm going to kind of just skip this because since your report was all over it, you should know all about what's going on right here. Okay? The only thing you may need to add to it is know what PCR is, which is what?
Protein chain reaction.
Yeah, polychain reaction. That's where we take the DNA and we duplicate it several million times so we have enough DNA to actually run the test with. The only other thing that I'm not sure if I told you, do you remember the liquid on top of the gel? Yeah, that was called TBE buffer. And for some reason that always pops up on the test. I don't know why. The second thing you'll need, last thing you'll need to know is how is the DNA strand separated. It's done two ways. It's one by size and then two by charge. Kind of remember that example I gave of the semi and the motorcycle going down the congested highway. Okay, the next several tables is And the next several tables is just evolution. That's lab number 19. I would make sure, if you haven't done it yet, take pictures, make drawings of everything on this table. Okay? For evolution. The next thing is these last two tables here with all of these skeletons of different animals. We're looking for homologous appendages. So essentially on every one of these skeletons you need to know what the humerus, the radius, the ulna, the carpals, metacarpals, and phalanges are. Now, for just about everything here, it's approximately the same. The only difference is on the frog. And on the frog's arm, you can still see the line here, right here. Because of the way that the frog jumps, it puts a lot of pressure on its front legs whenever it lands. So they ended up having to fuse their radius and ulna together so that there wasn't separation of the bone on landing. Okay? So this bone is called the radial ulna. And I'll tell you again, this one likes to make it on the practical a lot. What else? Oh, the last thing they want you to do is identify one adaptation on each of these skeletons that benefits the animal. Big thing I would say on the bat is its ability to fly. On the cat would probably be the addition of claws. And also, if you look at the cat's arm here, it actually walks pretty much on its tippy toes all the time. It doesn't actually put its wrist or its ankles down on the ground. For the bird, again, it's flight, and it's the difference of how the radius, ulna, carpals, metacarpals, and phalanges are put together on this one. That gives it the ability to fly, along with its feathers. For the turtle here, the big adaptation is carrying its house on its back. It's the addition of the carapace. Yeah, which one do y'all think evolved first, the top shell or the bottom shell? No, it was actually the bottom first.
That developed first?
Yeah. The reason it developed on the bottom first is because the early ancestors of the turtle actually swam at the top portion of the water, and it was actually getting hit from predators below. So this bottom carapace developed first, and then the top one developed later on. Yeah, if you're interested in that, look it up on the internet. It's a cool way that it evolved. Okay, and then the last thing for the frog is the ability to jump. All Okay, first thing is you need to know the different parts and pieces of the skull. If you have the review sheet here, it tells you all the pieces. If you need me to, I'll write them up on the board here a little bit later. My suggestion is start learning the parts from the colored skull here, but be able to do it on the plain colored skull, because they could put either or on the test. If they're nice, they put this one on there, it's easy to see. If they're not, they put this one on there, and then if you learned it on this one, you're kind of hosed when you get to this one. But the different ones you need to know is the frontal bone, the parietals, the occipital, the temporals, the sphenoid, which is the yellow here going all the way back, the zygomatic arch, the nasal bone, the mandible, and the maxilla. So mandible is jaw, maxilla is the top portion. And then the last thing is the foramen magnum, which is this hole right here where your spinal cord actually attaches. Okay, you'll need to be able to explain the differences in the frontal bone of the human and the frontal bone of both this is the chimp and this is the gorilla. And if you notice, in both the chimp and the gorilla, the frontal bone is almost gone. Okay, it's very much reduced here, so that reduces the frontal area of the brain inside the chimp and the gorilla. Now, one of the weird things is I was presented a study yesterday in one of the presentations in the other classes that actually show that chimp brain size from infant to adult pretty much peaks out, and by the time they're young adult, the brain stays the same size throughout the rest of their life. In human babies, the growth of the brain is pretty much exponential over the entire lifespan of the human. So that's probably why we have such big brains. Okay. Oh, second thing you're going to need to know is the difference in positioning of the foramen magnum. Okay, and how it influences either upright or four-legged position. And if you can see, the chimp's foramen magnum is pushed a little bit towards the back, or at least farther than the human's foramen magnum is. All right. Last thing is the PTC paper and the Hardy-Weinberg equation. Since we went over that the other day, I'm going to let that go. If you need more explanation, just let me know. But other than that, that is pretty much all of your test. It seems like there's a lot less on this test than there has been on the other two. So anyway, any questions on anything that we're... All right, I've got to give a little help, but then after that, I will get out my grade book